I'm very happy to be with you here today. And I'd like to tell you a little bit about our studies that focusing on how the microenvironment regulates vascular fate and assembly. In general, our lab is interested in understanding what regulates cellular fate to specialized cells, and then how the cells morph into functioning tissue. Specifically, we focus on the vasculature, where the vasculature grow and getting into the homeostasis state, but this homeostasis can be disrupted by various extracellular stresses that could happen during disease development. I listed some here, in injury and in extreme conditions. What we're trying to do in the lab is activate these morphogenesis and in some cases also fate pathways to develop therapeutic countermeasures to restore the function, repair, and towards homeostasis. So which stressors we study? We study low levels of oxygen, hypoxia. We study changes in the surrounding extracellular matrix, in flow and strain, and radiation. Today, I'd like to focus on a couple examples surrounding our work with hypoxia and with the extracellular matrix. So the first study done several years ago, we looked into how the differentiation or cellular or endothelial fate is impacted by hypoxia. Through the differentiation of our induced pluripotent stem cells through 12 days, back then it was about 12 days, we simply examine what happens when we do that in an atmospheric condition or in a hypoxic condition of 5%. And we notice when we culture the cells or differentiate the cells in 5%, we start to see these colonies and upregulation of expression of V-cadherin or vascular endothelial cadherin marker for endothelial cells. Nicely, we also notice that these colonies are V-cadherin positive cells that are surrounded by PDGF receptor beta positive cells, which are pericytes. With that, we wanted to understand, is it the early stages of differentiation that impacting the differentiation in hypoxia that impacting the fate or later on? So around the middle of the differentiation, around day 6, this is about a mesodermal induction. So we expose the cells to what we call secondary hypoxic or prime hypoxis, as well the control and continuous hypoxia. And what we have found is that when we expose the cells to either atmospheric condition or later stages of differentiation to hypoxia, we see no upregulation of specific endothelial marker V-cadherin and CD31. But we saw upregulation of these markers when we expose the cells during early stages of differentiation to hypoxia. We have done several other analysis where we show again upregulation of V-cadherin, CD31, and lectin, as well as the functionality of these cells. This study was published in 2014, but since then, what we notice in other published that I just not going to go over right now is that other stresses such as different stiffnesses of the matrices and also patterning of the extracellular matrix will impact this vascular fate. So with that in mind, that we know that hypoxia regulates the fate towards or impacting the fate towards a more endothelial cell, we wanted to ask how the hypoxia regulate vascular integration. Specifically, Bria Macklin, who was a PhD students in the lab back then, wanted to examine the hypoxic retina tissue. I'll explain a little bit later why we chose this specific hypoxic retina. So the first thing she did, she just compared the iPS-derived endothelial cells and mature human retina endothelial cells when she exposed them to hypoxic conditions, and we in vitro, and we saw no difference. She did notice that there is some difference in the proliferation rate of these cells in vitro, where we saw that the hypoxia did not impact the iPS-derived endothelial cells proliferation rate, but it did slow down their proliferation, the proliferation of the mature cells. She then moved to three-dimensional assay, where she took collagen gels and formed a network inside these collagen gels in hypoxia. and comparing again the IPS derivative to the mature retina endothelial cells. And she found that the hypoxic condition in the IPS case induced better and more networks compared to the retina mature endothelial cells. With that, we wanted to go in vivo and ask how we can study more about this, the integration of IPS cells in hypoxic conditions. For that, we used we collaborated with Elia Dow, who is at the Wilmer Institute at Hopkins, using this oxygen-induced retinopathy model, which is a classic to examine how our cells respond to hypoxia. During retinopathy, what's happening is that the healthy vasculature are exposed to a stress, in this case glucose, causing the capillary to drop out, this causing hypoxic or ischemic region in the retina, which induce pathological neovascularization. What we thought we could do is basically inject the cells in that point where we are generating this ischemic condition, hypoxic conditions, and see if they are going to stabilize the retina vasculature. So the way this assay goes, we take the PAPs on day 7, we expose them to, or we take them into high oxygen chambers, and then move them back to atmospheric condition, which induce the ischemia. And what we have found, and that when we inject the IPS-derived endothelial cells into this hypoxic retina, they integrate very nicely with the host vasculature. And you can see here in green are the injected cells, either the iPS cell derivative or the mature cells. And M-lectin is the mouse lectin. They integrate much nicer with the mouse vasculature, and you can see this clearly in these images where we see nice integration in yellow of the mouse and the human vasculature versus the mature retina endothelial cells that basically generating a layer of vascular network kind of on top of the mouse vasculature. So Bria continued to ask why this is happening. She used she came up with this nice assay where she encapsulated in the collagen gel SDF1 alpha, which will recruit endothelial cells, and basically seated the endothelial cells on top of this hydrogel, again, put them in hypoxic or atmospheric condition. As you can see here, that we found that the IPS derivative penetrated better to this collagen, either in atmospheric or hypoxic conditions. Indeed, she found in the hypoxic conditions that our IPS derivatives express the receptor for the SDF1 alpha, the CXCR4. And indeed, when we inhibited the CXCR4 using blocking antibody in vivo, when we injected that along with the cells, we see a much less integration of the human vasculature with the host vasculature. So all in all, what we're thinking is happening in the IPS derivative compared to the human retina endothelial cells, the IPS derivative proliferate faster, they're recruited to the host vasculature and integrate much more efficiently than the mature cells. So we're continuing the study to understand better also how or what is the role of the pericytes in these cascades, which are known to contribute to the retinopathy pathology. But in general, where we are thinking these studies or this area is going to, and I think Gordana gave a good overview of what we can do with these cells in large scales and also in organoids on the chip, is we're thinking it's going towards tissue-specific endothelial cells, where we'll be able to analyze how the endothelial cells impacting specific tissue or in the tissue environment that they're in, and vice versa, how the tissue environment regulate the functioning of these tissue-specific endothelial cells. And this will allow us to understand more about organ morphogenesis, develop more precise organ on a chip models as well as organoid, and hopefully develop a personalized medicine and drug discovery in specific organs in the body. So with that, we wanted to understand how hypoxia regulates vascular assembly.
And vascular assembly is happening in 3-dimensional. And towards that, instead of taking those collagen gels like what Bria did before and just put them in this hypoxic chamber and then take them out and analyze them, what we, Kyung Ming Park in the lab, who was a postdoc in the lab, developed this nice hydrogel system that is basically has a backbone. It could be a gelatin or polysaccharide such as dextran. And it's modulated with ferrolic acid that in the presence of laches, creating this reaction that consume oxygen. What's happening is then that we have hydrogels that while it's forming in the lab, it's becoming hypoxic. So we have hypoxic environment basically on the bench. When we add cells in those hydrogels, the cells continue to consume oxygen and those maintaining these hypoxic levels for longer periods of time. We found that it's with the endothelial cells or endothelial progenitor cells, they're continuing these hypoxic conditions up to 48 hours, which is enough to activate hypoxia-inducible factors 1 and 2, which takes about 4 to 6 hours to accumulate. So with this, I'm not going to get into too many details, but we encapsulated here endothelial colony forming cells. And as you can see here in the hypoxic condition, there was they promoted very nice vascular networks compared to the non-hypoxic, which kind of allow the cells to sprout, but not really form robust network. Through a series of studies, we found that the hypoxia, of course, stabilizing hypoxic inducible factor, in our case, both 1 and 2, and they upregulate the MMPs that allow the matrix to degrade, and then to the vascular bed to form. So with that in mind, this was basically our first study that demonstrated that there is a direct loop between a hypoxic condition impacting the matrix through their degradation. So with that, we thought about how the matrix mechanics regulate this vascular morphogenesis. And you know, there have been other studies that show the impact of stiffness on network formation. But we were actually interested in how viscoelasticity regulates vascular assembly. So most of our tissues in our body are basically have both elastic and viscous properties. This is a nice review published last year who's demonstrating the viscoelasticity properties of the matrix, their ability to respond to deformation by relaxing over time. So with that in mind, we wanted to understand is viscoelasticity regulate this morphogenesis of the vasculature. So Jai Wei and Rahel Schelleman in the lab developed this nice hydrogel system which composed from a dextran, so from polysaccharide and from gelatin, and we generated 2 types of hydrogel system. One with what we call dynamic cross-linking, which allow us to mimic viscoelastic properties, and one that is non-dynamic cross-linking. We made sure that we can decouple stiffness and stress relaxations in this hydrogel, so we can make the hydrogel soft or stiff and maintain that soft and stiffness throughout 3 days in culture. These are no cells here. And these soft or stiff materials can also differ in their stress relaxation. And this difference in stress relaxation can be maintained over 3 days of culture period. So with that, we wanted to understand how they modulate morphogenesis. So quickly, endothelial cells morph into vascular networks through a process that starts with vacuole or a small lumen formation. Then the cells then sprout and branch and form the network. And indeed, when we again encapsulated these cells
in the what we call the dynamic hydrogel, the viscoelastic hydrogel. They form these beautiful networks versus the non-dynamic hydrogel. We see that they are forming some network, but not as robust and complex as in the dynamic hydrogels. And you can see here a luminal structure forming in the dynamic hydrogels, which we cannot find in the non-dynamic hydrogel. We then wanted to understand more what is the mechanism that regulate this process. And we first examined if the vacuole and lumen formation process, which takes in culture about 4 to 6 hours, is modulated between the two hydrogel, and we did not find any difference. Both of them allow the cells to undergo vacuole and small lumen formation. Then we hypothesized that what's happening in our hydrogels is that the dynamic hydrogels allow the cells to contract and push and allowing them to sprout. And this does not happen in the non-dynamic hydrogels because they are not flexible. And indeed, by encapsulating fluorescence bead and following them with time lapse, we could see that the fluorescence beads moved while we have cells in the hydrogels move further and faster in the dynamic hydrogel compared to the non-dynamic hydrogels. And indeed, when we stand for a contractile marker, the phosphorylated MLC, myosin light chain, we could see that that the cells in the dynamic hydrogel are contractile. We also noticed that this contractility is leading to integrin clustering, and you can see that with basically clustering of integrin beta 1 in the dynamic and not in the non-dynamic. We also done some qPCR and showed the differences in integrin beta 1 and integrin alpha V in the dynamic versus the non-dynamic. This lead to large focal adhesion formation, here marked by vinculin. Again, that we could see the difference between the dynamic and non-dynamic. And when we inhibit the contractility of these cells with blebbistatin, we could see, of course, a downregulation of the integrin clustering, and this leads also to inhibition of network when we inhibit cell contractility compared to controls. We then looked into the activation or phosphorylation of focal adhesion kinases, and again, we could see upregulation in the dynamic hydrogel and the non-dynamic hydrogel. When we inhibit phosphorylation of focal adhesion kinases, we could see inhibition of the network compared to the control. Our cells not just sitting there and starting to make networks, they are actually interact with the matrix. They start to degrade the matrix, and they degrade it faster and better in the dynamic hydrogel. And we see that with expression of the membrane MT1 and MMP1 and MMP9. And again, when we inhibit with the general inhibitor of MMPs, we could see inhibition of network formation. Nicely, we could also see that when we inhibit the MMP, we could see that the degradation of the matrix that is seen here with the reduction in stiffness, it's slowing down with that inhibition. Nicely, these cells would also produce their own extracellular matrix, both collagen 4 and laminin. And you can see here very, very nicely in red, the collagen 4 is deposited around a luminal structure in the dynamic hydrogels. Finally, we also examined if the relevancy of this system for in vivo as we were thinking about therapeutics, and we injected them with the endothelial colony forming cells. We can see here network forming in the dynamic hydrogels, and here you see day 7, we see larger vessels in those hydrogels. And we can also see when we inject the cells, and this was done subcutaneously, when we inject these cells with no cells, we see that these hydrogels, inject the hydrogels, we see that they encourage angiogenesis by these CD31 positive cells in the dynamic hydrogels, which we could barely find in the non-dynamic hydrogel. And if we found them, they would be typically at the interface between the tissue and the hydrogels. By injecting to the tail vein either Evans blue or lectin, we also confirm the functionality of those angiogenic vessels. We concluded that the dynamic networks allow the cells to contract, and this activates integrin clustering, followed by focal adhesion activation, MMP expression, and also extracellular matrix deposition, and all of that allow for a morphogenesis to happen. And with that, I'm going to finish. I'm just going to summarize that we found that oxygen tension regulates vascular fate using a matrix remodeling, and it also regulates vascular integration with host vasculature, and that a hydrogel networks dynamic of viscoelasticity regulate vascular assembly. Just to thank all the people that done the work, I tried to mention throughout the presentation, of course, our collaborators and our funders.